Hi guys, and welcome back. Today we are going to talk about gene expression and apparently signal transduction. That'll come a little later, okay? On last week's show, we talked about DNA replication. So if you remember, our central dogma is what we started with last week: DNA to RNA to protein. And we had looked at the process of making DNA out of DNA. That was called replication. Here, we're going to look at the rest of this all together. That's called gene expression. We'll start by looking at transcription, and then translation, along with how signal transduction plays a role. So here we are taking a sequence of nucleotides, and we are turning it into a protein, from DNA to RNA to protein. And the unit of DNA that we read and that we turn into a protein is called a gene, right? Remember in our old analogy we said a gene is kind of like a word. So right here what I have for us is the partial, this is not all of the nucleotides, this is just some of them, the partial nucleotide sequence for the phosphofructokinase gene. If you remember, phosphofructokinase, he was involved in cell respiration. And in humans, our genes are quite long. In fact, the average human gene is about 150,000 nucleotides. The average protein in humans is about 300 amino acids. Remember, amino acids make up proteins. So obviously, somewhere along the way, we are doing a bit of a reduction. So we're going to see what happens there. Now, you do gene expression constantly. You do gene expression all throughout interphase, all throughout mitosis. You have to. And part of the reason is because you constantly have to regenerate those proteins that have been degraded. Your proteins do degrade at a pretty constant rate, so you have to replace those proteins. And you also want to make new proteins in response to your environment changing. So as an example, if you eat a candy bar, right, you want to make a whole bunch of enzymes that will help you to break down the sugars in that candy bar. So you have to be able to change your gene expression and regulate your gene expression, regulate what proteins you're going to produce. More on that in a little bit though. So here, in gene expression, here's sort of our outline. We are taking DNA and we are going to read our DNA, transcribe that DNA into a special type of RNA called mRNA. Remember, there's lots of different types of RNAs. mRNA is just one of them. The M here, by the way, stands for messenger. Okay. And then we are going to translate that mRNA into protein. So in this example, there's three genes right next to each other along a long molecule of DNA. We are just focusing on one gene. And notice we are just looking at one side. So it has highlighted here the DNA template strand. Now this term probably sounds familiar, the template strand, but it's not, I assure you. The term that you are thinking of is parent strand. And here's the thing. The template strand and the parent strand are not the same. They are not the same. The parent strand we use during DNA replication. The template strand we use during transcription. So be careful here, okay? Because it sounds like they could be interchangeable. So anyways, notice that we are just referring to one side of the DNA molecule as the template strand. Just the 3' to 5' side. Well, the reason why is because, as it turns out, we have to follow the same rules here as we do during DNA replication. We can only read, remember, we can only read DNA in one direction. We can only read it from 3 to 5. So we can only read one side of a DNA molecule. And here, it's the 3 to 5. Now, hypothetically speaking, what we could do is we can just read the bottom side backwards from right to left, 3 to 5, but eukaryotes don't really do that. Some viruses do, but as eukaryotes, we got plenty of DNA to go around. We just use one side. Seems like a waste, but it is what it is. Now, because we are reading 3 to 5, guess what? We have to write 5 to 3. And this goes not only for DNA, but for RNA. And I apologize, I keep hitting my next button. So this applies not only to DNA, but to RNA. So you'll notice our mRNA molecule here starts at 5, ends at 3. We had started writing it at this side, and went from left to right, from 5 to 3. Okay, so once we make our mRNA molecule, we translate our mRNA molecule. And the way we do that is we read 3 nucleotides at a time. We have a special term for those 3 nucleotides. We call them a codon. So in this example, UGG is a codon. U is the next codon. G is the next codon. Notice, by the way, my mRNA sequence is complementary to my DNA template strand. I had an A here, therefore I have a U here. Remember, U takes place of T in RNA. I have a C here, therefore I have a G here. C here, G here. A here, U here. A, U, C, G, etc. I'm still following my complementary base pairing rules. I'm still following my rules of reading and writing. Okay, going back to the codons. Each codon, we have 64 possible codons, will code for a unique amino acid. We have 20 different amino acids. And I know you're going 64 codons, 20 amino acids. The math doesn't quite work here. We'll see how that works in just a second. Transcription happens only in the nucleus of the cell. Makes sense? DNA is in the nucleus of the cell. Translation happens at the ribosomes. And where are the ribosomes located? Well, remember, the ribosomes are usually located on the surface of the rough ER. So transcription occurs in the nucleus. Translation largely occurs using ribosomes on the surface of the rough ER. Alright, just because you got a gene doesn't mean you have to use it. So we do use some genes more than others. As an example, here's gene A. As you can see, we are transcribing that guy a ton. Oop, I keep doing that. And therefore we are translating that guy a ton. Gene B, not so much. We don't need him as much. So we're not transcribing him as much. We're not really translating him as much. What regulates how often we read a gene is actually in part signal transduction. So do you remember way back when we had talked about cell signaling, and we had said that one of the things that cell signaling can trigger, and the process of signal transduction can trigger, is a change in gene expression. Well, here we are, gene expression. So we're going to bring that back in here. Okay, so a little bit more about regulating those genes. Some genes, by default, they are always on, right? Their default state is to constantly be expressed. Some genes are by default off. They aren't expressed all the time, or they're permanently shut off in a cell. So the genes that are always on, they actually do have a special term. They're called housekeeping genes, and I promise this is a real thing. You can Google it. Housekeeping genes. These genes are constantly expressed in all cell types. They are functional in all cells. You need them for just everything. You need them for cell respiration. You need them for transcription and translation itself, right? We need proteins to do this job, so we need those genes to be expressing those proteins. So all cells need them all the time. Their expression really isn't tightly regulated, if it is regulated at all. They're just on. So a couple examples. PFK. Again, going back to our buddy PFK. He's involved in glycolysis. Everybody has to eat. Everybody has to eat all the time. So everybody has to be doing glycolysis almost all the time, which means you got to be expressing PFK all the time. Almost everybody has microtubules that they constantly need to repair. So everyone is going to be expressing their microtubule proteins. Collagen, same thing. Everybody needs collagen. Everybody needs to make ribosomes too. About 1/10 of your genes, 2,000 out of 20,000, are these housekeeping genes. Most of them, though, are what are considered luxury genes. And luxury genes, that's not a technical term. That's a me term. I came up with that. So don't go, using this everywhere. These luxury genes are only functional in certain cell types. You only express them when you need them. And you only need them for certain cell activities. So, couple examples. White blood cells, in particular, produce antibody proteins. And it's only white blood cells that really produce those antibody proteins, so white blood cells are the only ones that are going to read those genes. Red blood cells are going to be the only ones who read the hemoglobin gene. Neurons are going to be the only ones who read their ion channel genes. So these are the vast majority of your genes. About 90 to 95% of your genes are these luxury genes. And because they are luxury, you can't always spend money on luxury, right? You have to highly regulate these genes. And how we regulate them is usually by peptide or steroid hormones. Those are coming back to bite us in the ass, aren't they? Okay, so let's look at how these luxury genes are turned on. We have seen this, or something like this image before, okay? Here's a steroid hormone. Remember, steroid hormones are lipids. Steroids are lipids, which means they can go straight through the cell membrane. They go straight through the cell membrane. They meet up with a receptor that's floating around inside the cell. That receptor and its attached hormone will travel inside the nucleus and attach itself to DNA. And previously we left it at this. We said, that's it, we're going to come back later, and here it is, it's later. Well, that receptor plus its hormone will actually help to regulate expression of certain genes by attaching itself to DNA, and it'll cause us to express different mRNAs and therefore different proteins. So this is how most luxury gene regulation takes place. Okay, let's take a look at the structure, the DNA structure of a gene now. Okay, this is the DNA structure of a gene. This is a bit of a simplified version. I know you look at this going, hell no, this isn't simplified, but I promise you it is simplified. So we have, within the gene, we have the area of the gene called the coding region. This coding region, let me erase my marks here. This coding region is what is ultimately turned into mRNA. It's what's ultimately is transcribed, I should say, into mRNA. In front of the coding region, we have regulatory regions. These regulatory regions help to determine when and where that gene is expressed. And if this is a housekeeping gene, those regulatory regions say on all the time. If this is a luxury gene, those regulatory regions will be more selective. And remember on the previous slide when we talked about the receptor and the hormone? Well, that receptor-hormone complex will actually attach itself to these regulatory regions. Receptor-hormone complex. Okay? Notice our regulatory regions, we don't make into mRNA. We don't transcribe those. Those are all behind the scenes. The real star of the show is the coding region. And notice our coding region, we can divide up into codons. So once we make our mRNA, remember it's complementary to our DNA, we will have nucleotides, and I'm just putting random nucleotides here, don't worry about the sequence in particular. Each nucleotide triplet, that's called a codon. And since the codons are in mRNA, they had to be in the DNA too. So we will take a double-stranded bit of DNA, like what we see up here, and a lot of times we'll reduce it down into these schematics, where we just see like a colored block or an annotated block. Keep in mind, we are seeing this does represent 2 DNA strands, and we only use one side. We only use the template strand. Okay, well, there's no way for us to tell the template strand here without 3 and 5, but we only use one side. But we often just reduce it down to this schematic. Okay, so all genes will contain certain things. Unsurprisingly, eukaryotes and prokaryotes, they differ a little bit. Let's look at what they have in common, and then we'll look at what's different. So all genes will contain what's called a coding sequence. A coding sequence. We already saw that. That's the part that's transcribed into mRNA. Coding sequence and coding region are actually the same thing. All genes will contain regulatory regions. In particular, there's one really important type of regulatory region called a promoter. So let me point this out. Do it in green here. Come on, there we go. These regulatory regions are composed of a couple dozen to a couple hundred nucleotides. A promoter is one of these regulatory regions. So I'm just going to say that this green chunk on the right is a promoter. We have other types of regulatory regions too, but the promoter is one of the more important ones, and all genes have a promoter. What's the job of the promoter? Well, as you can guess, it promotes. Okay? It promotes gene expression. Now within the coding sequence we also have codons. We saw this too. We have these codons. We really only pay attention to them when we're ready to do translation though. So we really only pay attention to them once we reach the mRNA stage. But they're there in the DNA too. We have to have somewhere to tell us where to start and stop transcription. So this mRNA, remember, it's just made from the coding region. We just transcribe the coding region. We don't transcribe the entire gene. So we have to have somewhere that tells us to start and somewhere that tells us to stop. So we have to have what's called a transcription start signal. Let me draw this in. It's not in my diagram right now. We have to have a transcription start signal. And a transcription stop signal. Let me clean up my stuff here. Here we go. Transcription stop. In addition to transcription start and stop, we also have to have a translation start and stop, right? Because, well, we're going to transcribe from all the way through the coding region, but once we make the mRNA from that coding region, we're going to translate it, right? So we have to have somewhere to tell us where to start translating. So where we start translating is a little bit after the transcription start, and that's actually what's called the start codon. So the start codon is the translation start. The stop codon is the translation stop. Okay, so start codon and translation start are the same thing. Transcription start is not the same as the start codon though. Be really, really careful with this. You do not start transcription at the start codon. You only start translation at the start codon, okay? Similarly, you do not stop transcription at the stop codon, you only stop translation. Let me try making this a little bit clearer. Let me make a neater mRNA molecule down here. Okay, so I'm going to make an mRNA molecule. Alright, this very first nucleotide on the mRNA molecule, I'm not going to say it's a particular nucleotide, I'm just going to say N. That was my transcription start. At the very, very, very end of the mRNA molecule, I had my transcription stop. And the transcription start and transcription stop nucleotides are actually going to differ from organism to organism. Everybody has slightly different versions. Not everybody, but eukaryotes and prokaryotes have slightly different versions. Now a little bit inwards from that transcription start, I just have some junk nucleotides that we don't use. A little bit inwards I have my start codon. I don't immediately have my start codon right after my transcription start. We won't get into why, but it's just a thing. And here's my stop codon. Remember, another name for start codon is translation start. Another name for stop codon is, switching the slide, is translation stop. So transcription and translation start and stop are not the same sequences. And then of course in between we have all of our other nucleotides. Okay, so everybody's got transcription start and stop signals. Everybody's got translation start and stop signals. Remember, translation start is the same as a start codon. Translation stop is the same as a stop codon. We always use the same start codon for every living thing. So in the DNA, my start codon is always going to be TAC. So if I transcribe that to RNA, it's always going to be T to A to U, C to G. My start codon is always going to be AUG. Stop codons are going to be a little bit, they can be a couple different varieties, a couple different flavors. Alright, so in addition, eukaryotic genes also contain additional regulatory regions. So see here how I have two green areas? Well, eukaryotes sometimes have an additional chunk of nucleotides next to the promoter that help us to fine-tune our gene expression. Sometimes we have 2 or 3 additional regulatory regions, or maybe even 4 or 5. And these regulatory regions, they have a couple different names. Let me give you some of those names. Let me draw it in green because that's the theme we're going for. So regulatory regions. In eukaryotes. Oops. Keep doing that. We have, of course, the promoter. Everybody's got a promoter. The promoter promotes. We often have what are called enhancers. We have what are called silencers. And we have a lot of others. Lots of others. Under a lot of different possible names. Okay. What we are really going to focus on are promoters, or are enhancers and silencers. Okay. So prokaryotes, they mostly just need promoters. Sometimes they have some additional regulatory regions, but not always. They're pretty simple. Prokaryote gene expression and gene regulation, they don't really need to worry too much about it. Eukaryotes, though, we do have to be very, very careful. The analogy I like to use here, and I think you guys have probably heard this, is prokaryotes are kind of like, right, they're usually single-celled, they tend to be a little simpler. Prokaryotes are kind of like that old hermit who lives in the woods. You know, that old hermit can shoot guns in his backyard, he can make noise, he can set off fireworks, but he doesn't have any neighbors for miles and miles around. He can make noise and do whatever he wants and not bug anybody else. So he doesn't have to very tightly regulate his behavior. Eukaryotic cells, on the other hand, are often multicellular. We've got 10 trillion cells inside us. And so I like to think of our cells as living in skyscrapers with very, very, very thin walls. You can't piss off your neighbors. So you have to very tightly control your behavior, and therefore you have to very tightly control what proteins you produce. So we have a lot more regulatory regions in our DNA to control our gene expression. So again, a promoter just promotes, all right? Helps to increase gene expression. Enhancers, well, they also, as you can guess, they also help to increase gene expression. They enhance. Silencers, man, these names just keep coming, right? They silence, they decrease gene expression. And we have a lot more besides those. And new ones are being discovered all the time. Okay, so eukaryotic genes contain enhancers, silencers, etc. They also contain what are called introns and exons. Introns and exons. So if you guys remember way back when we were talking about the structure of a genome, we said that less than 5% of our genome actually coded for genes, right? And we said that not only is there junk between our genes, there's also junk within our genes, too. So that junk, at least we think it might be junk, we're actually finding new functions for it all the time, but right now we're going to treat it as junk. That junk we call introns. Oops. The junk we call introns. The useful stuff that actually will help us to produce proteins we call exons. And keep in mind, with all genes, only one strand, the template strand, is transcribed at a time. And this is true whether you're a prokaryote or eukaryote. Okay, so that's the structure of a gene, right? Now at this point, imagine that we have transcribed that gene. We're good, we're done. We've made our mRNA. Okay? Let's take a look at the codons within the mRNA now. This is where we really start paying attention to the codons, even though they've been there the entire time. When we look at that coding region, we can split up the nucleotides in the coding region into these 3 nucleotide chunks called codons. Alright? The very first codon, where we start reading that codon and translating into an amino acid, that's called the start codon. That's where we start translation. Remember, it's translation now. The stop codon is where it's where we stop translation. Right here, this would have been where we maybe started transcription. So transcription start. This maybe would have been where we stopped transcription. Okay? So the start codon will cause us to place an amino acid. So start codon will code for an amino acid. The next regular codon, that'll also code for an amino acid. So will this one, so will this one, so will this one. All the way down the line, each one of these will cause us to link a new amino acid to a protein until we hit the stop codon. At the stop codon, that doesn't code for anything, any amino acids at all. We stop creating a protein at the stop codon. So how do we know which codons match up to which amino acids, right? Remember, we had 64 codons, 64 possible codons, but only 20 amino acids that we commonly use. Well, our cells have an internal codon table. And I like to use this version of the table the most. It's a radial table. You also see, traditional more column and row table. I think, a little too far. I think this one is a little bit easier to read. Let me go back and let me walk you through an example of how to read this. So this is our DNA, right? It has T's, therefore we know it's DNA. Let's transcribe it into RNA really quick. The T becomes A becomes U, C becomes G. That's our first codon. Let's look at our second codon in blue. C becomes G, G becomes C, A becomes U. Took me a second. So let's look at what AUG, what amino acid AUG codes for, our start codon codes for. AUG, methionine. So the first amino acid in our protein will be methionine. Let's look at GCU now. GCU, alanine. Our second amino acid will be alanine. Here's TTT, that will be transcribed to not UU. See, even I do it too, right? That'll be transcribed to AAA. So let's look at what AAA codes for. AAA, lysine. So the order of the nucleotides that's in our DNA becomes the order of the nucleotides in our RNA, and that becomes the type of amino acids and what order the amino acids are in our final protein. If we change one of these nucleotides, well, we may change what amino acid we put here. And that's how mutations work, by the way. We change what amino acids we put in the protein. Okay, keep in mind whenever you see these codon tables, they're almost always meant to be read off the mRNA. Alright, I don't think I've ever actually seen a codon table that's where you read off the DNA or tRNA. tRNA is another mRNA type of RNA we'll see later. Okay, so keep in mind with these codon tables, we always have to read them off the mRNA, which is why over here I had to transcribe into AUG and GCU, etc. Now what's really kind of interesting about this codon code, and don't worry, we'll get to the cats in just a second, okay? The codon code is actually considered degenerate. Degenerate. We have 64 possible codons, but only 20 amino acids. That means that multiple codons often code for one amino acid. Here, look at arginine as an example, right? We have 1, 2, 3, 4, 5, 6 different codons that code for arginine. So, and the reason why why we have 64 different codons, 4 possible nucleotides, AUCG, 3 nucleotides in a codon equals 64. 4 to the third equals 64. If a codon were only 2 nucleotides long, it'd be 4 squared equals 64. That wouldn't work, because we got 20 amino acids. But what that does mean is we do have to double up or triple up or quadruple up or even sextuple up on codons for each amino acid. So we call this doubling up or tripling up degeneracy. And it does help to guard against mutation, right? So as an example, look. If we have a CGU that we accidentally changed to a CGG instead, guess what? We're still good. Even though we did get a mutation, it doesn't change the final amino acid that we place in the protein. So that mutation essentially is silent. Doesn't affect anything. Some amino acids, like look here, methionine, only have one codon. That one kind of makes sense, right? It's the codon that we start with. That's the methionine that we start with. So it should only have one possible start signal. Tryptophan, for some reason, also has one codon. Here's all of our stop codons, by the way. Notice they don't code for any amino acids. They just tell us to stop translation. Now, the codon code is almost universal in all of life. So you can take a gene from one living thing, stick it into another living thing, and that gene will often be properly expressed. It'll be transcribed and translated most of the time. Now this is way easier if you're going from eukaryote to eukaryote, obviously because the genes between eukaryotes and prokaryotes are a little bit different. But we can take a eukaryotic gene and put it into prokaryotes, we just got to modify it a little bit. So what that means is a gene from one species can be properly expressed if placed into another species. This is what's happening with the cats. These cats have actually been genetically modified. Genetic modification is where you take a gene from one living thing and transfer it into another living thing. These cats have been genetically modified with a gene from jellyfish. It's the gene that causes the jellyfish to glow. So jellyfish have a gene that codes for a protein called GFP. Stands for green fluorescent protein. We also have orange fluorescent protein and red fluorescent protein and all those. So all we gotta do to make a glowing cat is stick that GFP gene or OFP gene into cats and they will glow. Easier said than done, but I mean that's the idea. So these are real pictures. I don't know about you, but I really want a glowing cat. Can't buy them, at least not yet. You can buy, in some states, you can actually buy glowing fish, though, that have been genetically modified. Okay, I'm going to pause here. I'm going to introduce the process of transcription itself in our next video. Thanks for sticking with me, as always.